a black background with white writing that says Georgia and Kat's accessible audio-described science videos with an exclamation mark at the end of the sentence. Underneath there are the words biology practical, DNA extraction. There are also 2 pictures that are of the structure of DNA, which is called a double helix, and 9 slices that are of a kiwi, which are green, and they are overlapping each other.
Hello and welcome to our accessible audio-described science videos. I'm Kat.
And I'm Georgia.
Today we're going to be extracting DNA from a kiwi.
On the screen there is a picture that is of all of the equipment which we are going to use for the experiment. There are scales, 2 glass bowls, a glass cup, a jug, washing up liquid, salt, a chopping board, a knife, a fork, 4 kiwis and a sieve. We are also going to use a spoon, but that isn't in the picture.
The first thing that we are going to do is measure out 25 grams of salt into a glass bowl. This is the first part that is for making a solution that will break down the cell membranes, which will allow the DNA to leave the cells.
There is a glass bowl that is on grey scales that have got a glass top. We are taking some salt out of a pot with a spoon, and then we are putting it into the glass bowl that is on the scales. We had to add it lots of times because of that it was difficult to put the right amount into it as the spoon is quite small.
The next thing that we're going to do is to add 80 grams of the washing up liquid to the salt.
We are squeezing washing up liquid out of a bottle into the glass bowl that has got the salt in it. The scales are set at 0 and we are putting the washing up liquid into the glass bowl until the scales say that there are 80 grams that are in there.
The last part that is for making this solution is to add the water into the glass bowl.
We are pouring 900 millilitres of water that is from a jug into the glass bowl. It is making the colour of the solution go peach.
Next, we are going to chop the kiwi into about 6 slices, and this will start to break down the cells.
We are now cutting the skin off of the kiwi, and then we are cutting it up with a knife into small slices before we are then slowly mashing it with the back of a fork until there are no lumps that are on there anymore, and then we are going to pour the kiwi slices into another glass bowl.
Now we've got to add 100 millilitres of the solution that we have made into the bowl that we've just put the kiwi into. The solution is now going to start to break down the membranes, but it could take a long time, so we're going to use heat to speed it up.
We have placed the bowl that has got the solution and the kiwi inside of it, inside another glass bowl that has got hot water inside of it. There is now a clock that has got its hands spinning around it, and the words 15 minutes later will come onto the screen after it. You will hear the sound of a clock ticking, and then you will hear an alarm siren sound.
Next, we are using a sieve to pour the solution into a glass cup so that we can have the liquid on its own.
There is quite a lot of the liquid that is going onto the table, as the sieve was larger than the glass, but we still had enough of it that was inside of it.
We had to wait for 15 minutes for the cell wall and the nucleus to break down, but we are now going to add some alcohol to make the DNA visible.
We are pouring some cold alcohol into a glass. As we are pouring it into the solution, the DNA is rising to the top of the glass. There are cloudy layers that are strands of DNA that are in between the layer of alcohol that is at the top of the glass and the kiwi. We are now going to hold on to the top of the spoon, and we are going to put the handle that is of the spoon inside of the glass and twist it to extract some of the DNA. There are now pictures that are on the screen that are of the layers of the DNA in different positions, including when that we had got some of it onto the handle that is of the spoon.
This practical is not a named GCSE core practical, but there are questions which come up frequently in the exams about it.
We hope that you have found this video accessible and interesting.
Please feel free to leave us a comment. And don't forget to subscribe for more accessible content.